Alright guys, welcome back to Sickle Cell Simplified, episode 2. Gonna dive right in. So today's episode is on what's called hemoglobin variants, and this is basically what determines how we have different types of sickle cell. When we look across the world, there are all different types of sickle cell, with hemoglobin SS being the most common, and then there's also hemoglobin Sβ thalassemia, hemoglobin Sβ thalassemia, hemoglobin SC, and also hemoglobin SO-Arab. There are some other different types of sickle cell, but for the purposes of this slideshow, we're only going to cover the top 5. As a reminder, when we're looking at our normal red blood cell, it carries hemoglobin A to transport oxygen through the body. Now we're going to zoom in on this hemoglobin A molecule to take a closer look at what exactly is going on with the hemoglobin. So each hemoglobin molecule is made of 4 protein chains. At the top, we have 2 alpha globin chains. These are important for the structure of hemoglobin. At the bottom, we have 2 beta globin chains. These are important for transporting the oxygen through the body. With no abnormalities, the body will just produce hemoglobin A. Any change, also known as a mutation, in the beta globin chains creates an abnormal hemoglobin. So the first change that we're going to talk about is the hemoglobin SS change. And here, instead of the patients having 2 beta globin chains, they actually have one hemoglobin S gene from one parent and another hemoglobin S gene from the other parent. Hemoglobin S is going to polymerize or sickle after it releases oxygen, and this is the most common type of sickle cell disease. Here we can see our alpha globin chains up top, our abnormal hemoglobin protein in the yellow, and our 2 S chains from each parent. These patients are going to have 100% of their hemoglobin being hemoglobin S. Let's take a look back at a normal red blood cell with regular hemoglobin A. To form this normal hemoglobin, beta chains are required. Any reduction in the beta chains is going to prevent red blood cells from being able to make that normal hemoglobin A. When beta chain production is reduced, this is called thalassemia. So there are 2 types of hemoglobin Sβ thalassemia. The first one I'll mention is hemoglobin Sβ +thalassemia. So here we have 2 alpha globin chains, our abnormal hemoglobin protein, and then we have 1 beta plus chain, and then we have another S chain. The other type of hemoglobin Sβ thalassemia is hemoglobin Sβ thalassemia. Very similar names, but here we're going to still have our 2 alpha globin chains, a hemoglobin protein, and then the difference is the beta thalassemia chain here is beta 0, and we still have our hemoglobin S chain. I'll leave this slide here for just a second so you can see both types of thalassemia. Again, on the left, in the blue box, we have our beta plus chain, and on the right, in the green box, we have our beta 0 chain. Let's take a closer look at hemoglobin Sβ +thalassemia. Here, the patients are going to inherit 1 hemoglobin S gene and 1 hemoglobin beta plus thalassemia gene. The hemoglobin Sβ +thalassemia allows for some production of hemoglobin A. In this type of thalassemia, I like to think of the plus sign as plus some hemoglobin A. The presence of some hemoglobin A reduces the hemoglobin S polymerization, also known as sickling. Disease severity for these types of patients is typically more mild than a hemoglobin SS patient or a hemoglobin Sβ0 thalassemia patient. Because these patients have some production of hemoglobin A, typically it brings down the percentage of hemoglobin S. A typical patient with hemoglobin Sβ +thalassemia would have a hemoglobin S percentage about 70 with a hemoglobin A percentage of about 30. Let's now take a look at hemoglobin Sβ0 thalassemia. Here, these patients inherit 1 hemoglobin S gene from one parent and 1 hemoglobin beta 0 thalassemia gene from the other parent. The hemoglobin beta 0 thalassemia allows for little to no production of hemoglobin A. In this type of thalassemia, I like to think of the 0 as 0 production of hemoglobin A. Because these patients have little to no production of hemoglobin A, they typically have a high rate of polymerization or sickling. Disease severity is typically as severe as hemoglobin SS. I'll put this slide up for just a second so you can see the comparison between the hemoglobin Sβ plus thalassemia, where we get a little bit of that hemoglobin A to bring down some of our hemoglobin S levels, and then our hemoglobin Sβ⁰ thalassemia, where we get little to no production of that hemoglobin A, so the hemoglobin S percentage remains fairly high. The next type of sickle cell disease we'll talk about is hemoglobin SC disease. Here, the patients inherit one hemoglobin S gene from one parent and one hemoglobin C gene from another parent. Typically, hemoglobin SC is a little bit milder, but it can still have some serious complications. An interesting thing that happens in hemoglobin SC disease is that yellow hemoglobin molecule actually turns into a crystal whenever it releases oxygen. We actually end up seeing more sickle cell eye disease, also known as retinopathy, and hemoglobin SC disease. I'm unsure if this is due to the crystallization of the hemoglobin molecule, but it's very interesting nonetheless. All in all, in a hemoglobin SC patient, about 50% of their blood is going to be hemoglobin S, and 50% of their blood is going to be hemoglobin C. Lastly, we'll talk about hemoglobin SO-Arab disease. I haven't seen a ton of this type of sickle cell, but it definitely is out there. Here, the patients inherit one hemoglobin S gene from one parent and another hemoglobin O-Arab gene from the other parent. This type of sickle cell has polymerization, or sickling, similar to a hemoglobin SS patient. These patients can still experience pain crisis, anemia, and other complications of sickle cell. These patients are going to have 50% of their blood being hemoglobin S and the other 50% of their blood being hemoglobin O-Arab. I want to do a quick recap of all the different types of hemoglobin. First, we have our normal hemoglobin with 2 alpha chains and 2 beta chains. Here we have no production of hemoglobin S and 100% production of hemoglobin A. Next is hemoglobin SS, and here we have our 2 alpha chains and 1 S chain from one parent, 1 S chain from the other parent. These patients make 100% hemoglobin S and 0% hemoglobin A. Next is hemoglobin Sβ plus thalassemia. Here, the patients are going to inherit one of those S chains and one of the beta plus chains. Because we do have the beta plus chain, we will see some of that hemoglobin A. These patients have 70% hemoglobin S and about 30% hemoglobin A. And we have our hemoglobin Sβ⁰ thalassemia. Here, the patients have 2 alpha chains and they have the S chain and a beta-0 chain. These patients are going to produce 100% hemoglobin S and no hemoglobin A. Next is hemoglobin SC. These patients have 2 alpha globin chains. They have the hemoglobin S and the hemoglobin C. And again, their hemoglobin is the one that forms into the crystal after it deoxygenates. These patients are going to have 50% hemoglobin S and 50% hemoglobin C. And lastly is the hemoglobin SO-Arab. We still have our 2 alpha chains, and then we have an S from one parent and an O-Arab gene from the other parent. Here, the patients are going to make 50% hemoglobin S and 50% hemoglobin O-Arab. Well, that wraps us up for today. I appreciate you guys and your time. Thanks so much, and we will be on the next episode soon.